Hello, I'm Bodo Grimbacher from the CCI in Freiburg in Germany, and together with my co-chair, Professor Eleonora Gambineri from Florence, I would like to welcome you to the PID and IBD session of the ESID 2020 meeting. The topic of gut infection and gut inflammation is very important to our daily work with our patients, as I guess many of the unsolved questions in our field are Paris, who will fill us in on the novel genetic causes of IBD. Professor in the gastrointestinal commensal bacterial. Kate is head of the
Well, first, I would like to thank the organizer to have invited me to discuss known and novel PID underlying very early onset inflammatory bowel disease. So I have no interest to disclose. So as you know, the gastrointestinal tract is one of our main body interface. It's a place we are constantly exposed to multiple antigens, notably of dietary origins in the upper part, and to a complex and considerable microbiota in the lower part. And we are also regularly confronted with pathogens. So this gastrointestinal tract has evolved as a highly regulated barrier where a strong, complex partnership between epithelial cells and immune systems set up a state of physiological inflammations, which is necessary to maintain intestinal homeostasis and to preserve the function of digestion and absorptions while avoiding pathological inflammation. We know from studies of knockout in mice that multiple genes are involved necessary to the control of this barrier, and we are now learning from human defect that at least more than 100 genes are necessary to maintain this barrier and to avoid, for some of them, to avoid pathological inflammation. So what I would like to do in this talk is first to define which genes effects impair the gut barrier and lead to intestinal inflammation of very early onset, which is by definition before 6 years. To discuss what are the individual consequences on clinical presentations and what means exactly Vero IBD, and I will try to attempt some classifications. And finally, in the last few slides, I will discuss whether all Vero IBD are of monogenic origin. So as an attempt of classification, I thought it could be useful perhaps to discuss separately the disease which affect one on the other partner of the gut barrier, the epithelium or the immune system. So let's first start by the disease affecting the epithelium. Not all of them are clearly PID, but some of them can mimic Vero IBD and they need to be known. So most of them are, we call them congenital diarrhea because they start very early after birth by a severe diarrhea. Most of them are not associated with inflammation, yet some of them are. And it's notably the case of three types of disease. Two diseases which affect very severely the differentiation of the epithelium. The first is microvillus inclusion disease, where mutations in 3 genes lead to a severe defect in the development of the brush border and loss of apical polarity. And a disease called tufting disease, where mutations in 2 genes result in very severe defective epithelial adhesions. So in both cases, there is a severe defect in epithelial entantiety, leading likely to bacterial translocations and exposing to inflammations, which can sometimes mimic VEO IBD. Apical transporters are rarely responsible for inflammations, except some of them responsible for chloride diarrheas, which, for reasons that we do not understand completely, can be associated with severe inflammation. So besides this disease, which are more epithelial defect, we have some defects in epithelial innate functions, which can lead to colitis. And indeed, we have recently described 2 such diseases. A first disease which is associated to a defect in a highly conserved enzyme called DUOX2, which is an NADPH oxidase necessary expressed by the gut epithelium and necessary to produce hydrogen peroxide, which is a highly microbial. And likely the lack of this enzyme results in poor containment of the microbiota and inflammations. I must say that this child in which we have described these disorders has been much better after a few years, suggesting that it's more a predisposing genes. Along the same line, we have also recently described a defect in intestinal alkaline phosphatase, illustrating the role of the epithelium in regulating the innate defense. Here, this enzyme, which is also highly conserved, is produced in the small intestine, released in the lumen at the surface of vesicles, where it can act on LPS, removing a phosphate in the lipid moiety, which thereby reducing by 1,000-fold the agonist activity of LPS on TLR4. And indeed, in mice, when the lack of this enzyme lead to a severe colitis TLR dependent. And we have indeed observed with Alexio Buiz such colitis or severe colitis in 2 children with loss of function mutations in this enzyme. In this specific case, perhaps we could have treated these patients with the recombinant IIP, but unfortunately they had surgery before that. So beside this disease, which affect mainly the epithelium, we have now disease which affect both the immune system. In some cases, it's congenital diarrhea, again, a severe defect of the epithelium associated with an immunodeficiency. And we have here 2 very emblematic diseases, trichohepatoenteric syndrome, where mutations in 2 genes which belong to the SCY complex involved in mRNA decay lead, for reasons that we do not understand well, to a severe epithelial dysfunctions affecting gut and liver, immune deficiency, dysmorphia, and typical hair abnormalities. And the second defect in the genes called TT7A, which can lead in the gut to multiple atresia or extremely severe enterocolitis, but at the same time to a complex and variable immunodeficiency with thymus alterations. The mechanism is not completely understood and related to the scaffold functions of TT7, which can associate with many proteins at the membrane, in the cytosol and in nuclei, and it's at least in the intestines probably responsible for severe epithelial apoptosis and loss of apical polarity, against resulting in loss of potentiality. So beside this disease, again affecting the differentiation of the epithelium, we have again deficiencies which are responsible for an immunodeficiency affecting the immune cells, but also the innate immune functions of the epithelium. And it's particularly the case of a defect that you know very well, the nemo-hypomorphic mutations, where the immunodeficiencies is associated with a defective NF-kappa B activations downstream many immune receptors. And there is also a severe colitis, which seems to be intrinsic to the epithelium as non-cured by hematopoietic stem cell transplantations. And a very nice paper published in Nature suggests indeed that the lack of NEMO results in increased apoptosis and loss of productions of defensins, which are necessary to contain the microbiota and avoid bacterial translocations, stimulating inflammation. So I had the temptations to associate to this defect mutations in RIP1 and caspase-8, which has been recently associated to a severe immunodeficiency associated with colitis. In fact, here, if the immunodeficiency is associated to a defect in NF-kappa B, it's not so clear that the intestinal inflammation is intrinsic to the epithelium, at least in humans. It's the case in mice, where there is a necropoptosis of the epithelium. In humans, it seems to be more monocyte necroptosis leading to inflammasome activations. So it should be seen probably as defect affecting the immune system, as together with the defect that we are now going to see. we are going to turn to the disease which affect the immune system predominantly, either because the gene is expressed exclusively in the immune system or because its function is mainly in the immune system. And here I would like to argue that the clinical presentations will be different. We will have two types of clinical presentations. One which affect mainly the upper part of the intestine, and some who affect predominantly the lower part of the intestine. And we have first, as a first type of presentations, what I would call autoimmune enteropathy with villus atrophy, lymphocyte infiltrate, often associated to allergy, notably food allergy, and to autoimmunity with cytopenia and endocrinopathy. These patients can develop colitis, but in this case, it's lymphocytic colitis and Jevesh-like colitis. And we have another group of patients where the presentation is dominated by colitis and enterocolitis, associated often with ulcerations and perianal lesions. And why I say so, it's based in fact on our own experience in our pediatric cohort, where in our cohort of more than 500 patients using TNGS and whole exome sequencing with Fabienne Charbiterion, with a remarkable pediatricians and geneticists, we see that we can classify our patients in three groups. Patients with epithelial defect, I already discussed that. Patients with autoimmune enteropathy, where when we look at the type of defect that we see, it's mainly defect impairing the adaptive immune system. And conversely, patients with colitis, where the genes that we found are mainly associated with the innate immune systems. So let's now try to discuss this starting first by the presentation with autoimmune enteropathy. So here, the yield of diagnosis is relatively high, 60%, not as close as what we see in congenital diarrhea, but still we probably know here a lot of molecular defects. And this molecular defect, as I just said, are defects which really impair the balance between regulatory T cells and effector T cells. This can be sometimes happen in the context of a combined immunodeficiencies, or it can be genes also known to be associated with common variable immunodeficiencies. But in our cohort of young children, it's really the disbalance between regulatory and effector T cell which seems to be important. And I will try to summarize the genes on these slides, knowing that I'm certainly not exhaustive, but I think that we should divide this defect in diseases which affect predominantly the Treg, with the emblematic FOXP3 deficiencies, which are manifest by autoimmunity, often associated with allergy. And I would put in the same category defect in IL-2 receptor A, STAT5, MALT1 and DOC8, where it's a little bit more combined with more complex immunodeficiencies. By oppositions with defect which affect mainly the effector T cells, enhancing their activations. And it's mainly the defect, the disease which are associated with enhancement of the JAK-STAT pathway with gain of function mutation in STAT1, STAT3, and as recently described, JAK1. And I'm just going to show you also a new defect in PTPN2. And we have defect in LRBA and CTLA4, which certainly affect the functions of Treg, but which probably also tend to enhance the functions of effector cells. So just a word about this new defect that we have just recently described with PTPN2. It's the case of a young baby starting a diary at 3 months with, as you see here, a very intense lymphoid infiltrate of its intestine, which resists to all type of immunosuppressive therapy until she received sirolimus, which was actually helpful before we made the molecular diagnosis. She had anti-harmonine antibodies. The Treg were normal. She had also normal IgE, and the TNGS was negative, so we went to exome to find, and I will not show all the data, but what we found is the missense mutations, which changed the cysteine, which is absolutely indispensable in the catalytic site of PTPN2, which is a phosphatase. And without this cysteine, in fact, the catalytic activity of the enzyme is totally dead, as we have shown with Fernando Lima. And so what is PTPN2? PTPN2 is a tyrosine phosphatase, which is well known in mouse and human lymphocytes to regulate JAK-STAT signaling. It can dephosphorylate the JAK1. It can dephosphorylate STAT, and notably STAT3. And indeed, when we look in the cells of the patients, here B cells treated with IL-21, you see the enhanced phosphorylations of phosphostat1, phosphostat3, and also, surprisingly, this phosphorylation of phosphostat5. So I will not go further into this defect, just to say that all the mutations involved in the JAK-STAT pathway are interesting to identify because they are probably accessible to a targeted treatment with JAK inhibitors. We have ourselves shown in patients with severe enterocolitis with STAT3 gain-of-function mutations resistance to all treatment, the efficacy of ruxolitinib. And recently, in this very recent paper just published in Immunity, Gruber et al. Insist on the interest of tofacitinib in the JAK1 gain-of-functions because they underline the fact that there is some kind of illegitimate phosphorylations of the JAK-STAT pathway beside beyond JAK1, and it's probably better to treat with tofacitinib, which is something probably to consider. So let's now turn to the disease which affect mainly the lower part of the intestine, manifesting with colitis of enterocolitis, with bloody diarrhea, very often granuloma and ulcerations, and eventually perianal lesions. And you see here that the yield of molecular diagnostic drop, perhaps less when we have colitis with perianal lesions, which are the most severe cases, but which drop seriously when we arrive to colitis only. So what do we have in this disease? In fact, we have, as you will see in the next slides, mainly defect affecting innate immunity and probably impairing in priority host microbiota interactions. And this is notably the case for these two types of defects. There is here the first very emblematic loss of regulations of colonic macrophages by IL-10 with the loss of functions in IL-10 receptors or in the productions of IL-10. And thanks to the work of Snapper and Jung, it's now clear that it's really the regulation of the colonic macrophage which is in front of the macrobiota which is impaired. And we have this defect which likely affect the bacterial clearance, as suggested for XIAP, TRIM22, and MPC1 by Ulik-Holm, and probably also the defect which affect the NADPH oxidase functions, NCF1, CIB, CIBA, and NCF4. Here, which are very often associated with intestinal inflammation, probably 60% of the case, by mechanisms which are not completely understood, which have been somehow associated with hyperproductions of IL-1 beta, but it remains quite unclear. So this actually leads us to another type of disease which we see associated with colitis, which are the defect of the regulation of inflammasome, with notably the gain of functions in NLRC4 associated with hyperproductions of IL-10, which can in very young children lead to neonatal enterocolitis with macrophage activation syndrome. Some specific case of mevalonate kinase deficiencies which have been associated in the neonatal period with severe enterocolitis. In that case, it's hyperproductions of IL-1. And I guess that we should add to these cases the recent mutations described in RIP1 and caspase-8, where necroptosis of the monocytes is associated with hyperproductions of IL-1 beta. And I should add to that defect in regulation of NF-kappa-B with the haploinsufficiency in A20, which can result in severe enterocolitis, and the recently described defect in TGF-beta1, which led to a very severe colitis associated with an encephalitis which was not completely understood. I must say that for the defect in the TGF-beta, I have not indicated here, but we have some other defects downstream the TGF-beta receptor, which can be also associated with more autoimmune-like disease of the upper part of the intestine. So I think that I have answered the first two questions that I wanted to address. And I would like, before conclude, to discuss whether all VO or IBD are of monogenic origin. And indeed, as I already mentioned, in patients with colitis only, the yield of diagnosis is relatively low. So it's possible that we do not know all the genes, but it's also possible that this type of colitis are also induced or dependent of the exposures to environmental factors. And I think that we should remember that we are now facing a very strong increase in IBD in children, exemplified here in these epidemiological studies published already 3 years ago, mainly in teenagers, but that we can also recapitulate at all age in this disease, in this data collected from the health insurance disease in France. And we know from and it's going to be discussed by Ari Sokol now, how the change in the microbiota likely play a role in the recent epidemics, or in the epidemics that we have seen now for a number of years in IBD in adults. And it's tempting to believe that these changes are now occurring in children, leading to this early onset IBD and also probably increasing disease severity. And this hypothesis is sustained by a very recent elegant work performed by the group of Gérard Rebel, who showed in mice that if you treat very young mice before weaning by antibiotics to reduce the microbiota, in fact, you make these mice extremely sensitive to the occurrence of colitis later in life, suggesting that indeed the recent change that we see in the microbiota could indeed promote early inflammations. So I will stop here and thank the people who have contributed to some of the data that I've shown you, besides the data of the literature, obviously. In the lab, I've already mentioned Fabienne. You have seen the picture of Mariana, who has been instrumental for a number of these studies. Frank Rummeler, who is the gastroenterologue pediatricians with whom we do the cohort. We have benefited from very nice collaborations with a number of people. Ulla Klaus in Dublin, Alexio Muiz in Toronto, Marina Alloy and Claudia Romano in Italy, Fernando Lima, and more recently Ken Botstuk. We have also important collaborations within the Institute Imagine, where I work, with Frédéric Rioloca and Sylvain Latour. And I'm going to stop here and to thank you.
So, hi everyone, I'm Ari Sokol, I'm gastroenterologist in Saint-Antoine Hospital in Paris, and I'm going to discuss with you the role of the gut microbiome as a friend and a foe. So here are my disclosures. So first, what is the gut microbiota? So roughly for an adult of 70 kilograms, you have about 2 kilograms of intestinal content. Of course, this is not only bacteria, there are many other things, but globally it represents 2 kilograms. We know that the number of microorganisms in our gut is huge, with roughly the same number of microorganisms in the gut than the number of cells in our body, so 10 to the 13. And for example, in 1 gram of stool, you have more than 100 billions of microorganisms. In our cells, we have a human genome, and of course, if everything is okay, we have the same genome in all our cells. If you put together all the genes present in our gut microbiota, it represents between 100 and 150-fold the human genome, so suggesting huge potential in terms of function. The microbiome is involved in many important physiological processes. I'm not going to go through them. Metabolism, immune function, intestinal barrier, protection against pathogen, and even the way our brain is functioning. In the gut, we have bacteria, and this is the most well-known part of the gut microbiota. And among these bacteria, particularly in the colon, there is mostly anaerobic bacteria belonging to these two major phyla, the Bacteroidetes and the Firmicutes. But we also know now that there are other types of microorganisms, such as fungi, but also viruses, particularly phages, that are viruses targeting the bacteria, and even more microorganisms such as archaea and protists. So in the gut, there is a very specific interaction, very specific relationship between our cells, the host cells, and the microbiota. And on the host part, you have the epithelial cells, but also the immune cells, and on the microbiota part, all the components I described. Interestingly, these interactions are going both ways. So our host cells are impacting the microbiota, and this can be in one part, and we're going to discuss that, driven by our genetics, and the effectors are host molecules, such as bile acids, but also antimicrobial peptides, mucus, and the inflammatory process itself, actually. But the microbiota also has important effect on our cells, on our physiology, and this is notably mediated by metabolites, but also microbial component present on the microbes. In some cases, so for example, in inflammatory bowel disease, in IBD, this interaction can be altered, and this will have impact both on the microbiota side and on the host side. What is particularly interesting with the gut microbiome is that it is under a double influence, the influence of our cells and also our genetics, and under the influence of environmental factors. And when you are talking about environmental factors, you can basically distinguish two periods in the life. First, you have everything that is happening early in life, and that can have some effect on the way the gut microbiome is going to interact with us at the beginning, so very early in life, because of course, in utero, we do not have any gut microbiota. There is no live microbiota. And there are many parameters that can have some impact. The way you are, the way of birth, the vaginal delivery or C-section, the type of diet, exposure to antibiotics, and also very simply the way you are living. It's really not the same in terms of microbial environment to live in a farm or in the middle of a big city. And now we have many epidemiological data showing that the perturbation of this first host microbe interaction early in life can have some impact on the immune system imprinting, and might actually lead to a kind of miseducation of the immune system. And this is supported by the fact that all this type of perturbation of the microbiome host interaction early in life are associated with an increased risk of immune-related disease, such as allergy, but also inflammatory bowel disease. So these are only epidemiological data, so only correlation, association, but we also have now a lot of experimental data suggesting this is actually true. And for example, there is this very nice paper from the team of Gérard Eberle showing that in mice model, perturbation of the gut microbiota at weaning induce some perturbation in the immune system education, leading to an increased susceptibility to intestinal inflammation much later in life. Of course, so this is the first period of life, but not everything is done at this early phase, there is adulthood, and during the adulthood, many other parameters can disturb the gut microbiota, the type of diet, exposure of course to antibiotics or to other types of drug, and also exposure to other types of substances such as dietary additives, such as emulsifiers. And again, any perturbation of the gut microbiota will induce perturbed function, and it can have also consequences on the host. To add another layer of complexity, all these processes, all these phenomena are modulated by the host genetics. And also, quite interestingly, I think there is potential effect on the next generation, on the offspring, because we know there is kind of transmission of the gut microbiome from the mother to the newborn. And if the mother already have an altered microbiome, it can be transmitted in a way to the next generation. It's clearly shown in animals, and there are many data suggesting this is the same in humans. So what about primary immunodeficiency? In this case, there is usually a clear genetic defect, and we can ask whether the gut microbiome could play a role in the phenotype, because the classical vision is that, yes, it's a genetic defect, and the consequences are all, the phenotypes, the clinical symptoms are all related to this genetic defect directly. However, it might not be so simple for all the type of manifestation. And I'm just going to give you one example based on our work on this gene CARD9, which is a gene encoding for an adaptor protein, which is expressed mostly in antigen-presenting cells and integrating signal downstream of many innate immunity receptors. So we studied this gene because CARD9 is also an IBD susceptibility gene, and we used CARD9 knockout mice submitted to this very common colitis model elicited by dextrosodium sulfate, DSS. And what you can see here is that wild-type mice in white will recover following the inflammation, whereas CARD9 knockout mice will not recover, and eventually, if you do nothing, they will die. So there is a kind of impaired recovery following inflammation in these CARD9 knockout mice. And we, as this adapter is important for signaling downstream of innate immunity receptor, we looked at the gut microbiota in these mice and observed that indeed CARD9 knockout mice had an altered gut microbiota, both regarding bacteria and fungi. So of course, this is only correlation, association, and all the interest to work with mice is to try to move to causation. So we did a first experiment, which is actually not published yet, based on germ-free animals. We used germ-free wild-type mice and germ-free CARD9 knockout mice we generated, and we colonized simultaneously all these mice with the same wild-type microbiota. And we analyzed the microbiota sequence at different time points. So this is the microbiota of all these mice 7 days after colonization. Each dot represents one mice, and you can see that basically after 7 days, all the mice are together. You can see that 2 weeks after colonization, there is some kind of shift, and 3 weeks after colonization, there is a clear shift and a difference between the mice, the wild type, and the CARD9 knockout mice colonized by the same microbiota. So there is this experiment clearly shows that the host genetics can have some effect on the microbiota composition. Here, it's only based on composition. So then we wondered whether this altered microbiota in these mice could play a role in the phenotype we saw, which is an increased susceptibility to intestinal inflammation. So Bruno Lamas, who was postdoc at this time in the lab, did the following experiment. He colonized wild-type germ-free mice with either normal microbiota from wild-type mice or microbiota from Cas9 knockout mice. And so we got two groups of mice. All of them were genetically normal. One of them had a normal microbiota in blue, and the other group had an abnormal microbiota from Cas9 knockout mice. And so all these mice were then submitted to this DSS colitis model. And what we saw is that by transferring the microbiota, we basically transfer the phenotype. Mice colonized with a knockout microbiota, if you can call it like this, had a higher susceptibility to colitis. You can see this in terms of weight, but it's the same in terms of histological scoring and all the other parameters we looked at. So I'm not going to dig into the mechanism, but you can look at this paper we published in Nature Medicine a few years ago if you want to know more. But this is a clear result showing that the host genetics, and in this case Cas9, can play a role on the modulation of the gut microbiota composition, but more importantly, function. And in the case of this study, by removing Cas9, actually we induce the generation of an altered microbiota with an altered function, and that leads to a higher susceptibility to inflammation. Of course, this does not rule out the fact that the genetic effect by itself can have, and of course has some effect, some direct effect, independently of the gut microbiota on a phenotype, and of course it might be also true for the intestinal phenotype. So then we can ask, okay, we have this data, this interesting data in mice, but do we have any data suggesting that this is also true in human, that the gut microbiota can be an actor in a phenotype, and in this case, in the intestinal inflammation pathogenesis? So I think that now we clearly have this answer, and the pragmatic answer is yes. And this is based on inflammatory bowel disease. We know for quite a long time now that the patients with inflammatory bowel disease have an abnormal microbiota, and this is just one example of the many studies that have been published showing the abnormal microbiota in IBD. So on the left part of the slide, you can see what we call beta diversity. So each dot represents one sample, and if two dots are close, it means that the microbiota are similar. If they are far, it means that they are different. You can clearly see that the healthy subject in blue have a different microbiota compared to the patients with IBD in flare, and patients in remission are basically overlapping between these two groups. So there is an abnormal microbiota in terms of composition. There is also an abnormal microbiota in terms of diversity. You can see on the top right of the slide that there is a decreased diversity of the microbiota in patients with IBD. And in terms of composition, on the bottom right, you can see that patients with IBD have a group of bacteria that is blooming in orange, and these bacteria are proteobacteria, are the bacteria from the proteobacteria phylum. And among this phylum, you can find notably E. coli. So besides the abnormal microbiota composition, which is nice but not proof of the effect of the microbiota, we know we have many data in mice showing that by modifying the microbiota, you impact the disease severity. And this is also true in human. And of course, this is more important, notably thanks to the few fecal microbiota transplantation studies that have been done, including the one we performed ourselves recently and published in Crohn's disease. Finally, there are now also many papers showing that the microbiota of patients with IBD have pro-inflammatory effect by itself. So in this very nice study published last year, for example, it has been shown that if you take the fecal microbiota from patients with IBD and transfer it in germ-free mice, it will basically exacerbate the colitis severity compared to mice colonized with the microbiota of healthy donors. So clearly demonstrating that there is pro-inflammatory effect of this microbiota from IBD patients. So, of course, this is IBD. This is not PID. So we can ask how to interrogate the role of host genetics in the modulation of the gut microbiota in human in the context of primary immunodeficiency. So we try to address this question through a collaboration with Alain Fischer in Imagine Necker Hospital in Paris. By doing this analysis, we analyze the fecal microbiota of patients with 3 types of rare monogenic PID causing IBD-like condition. So patient with chronic granulomatous disease, CGD, CIAP deficiency, or TTC7A deficiency. So I will not go into the detail of this deficiency, but all these deficiencies are inducing very different types of abnormalities on the immune response. So they are different diseases with different types of consequences on the immune system. So when we analyze the microbiota of all these patients and also of healthy controls and patients with conventional IBD, we were very surprised to see that basically the patients were clustering according to their genotypes, to their disease. You can see that the patients with CGD are clustering together, patients with CIAP deficiency are clustering together, and patients with TTC7A deficiency are also clustering together, showing that there is an association between the genotype and the microbiota composition. And when we looked at the precise characteristics of the gut microbiota of this patient, compared to healthy subjects, we observed many differences, and these differences were, again, specific of each genotype. So, for example, here you can see that in patients with XAP deficiencies, there was an increase of many bacteria that are normally found in the oral cavity. This is the case for Veillonella, for Fusobacterium, Scardovia, Rothia dentocariosas, which are really normally found in the mouth. Interestingly, also we found some very rare bacteria in patients with CIAP deficiency, particularly this Lactococcus garviae, which is known to be a pathogen of saltwater fish. And in all these cohorts, we found this bacteria only in the patients with CIAP deficiency, again suggesting a quite specific association between the genotype and the microbiota. On the other hand, for example, with patients with TTC7A deficiency, we found an increase in many bacteria from the Proteobacteria phylum, and patients with CGD had other specific alteration. So I think this is very interesting because all these data suggest that the alteration in the gut microbiota composition is not only a non-specific feature of intestinal inflammation. It's not only a consequences of intestinal inflammation, but it suggests that host gene might also actively modulate this process. And actually, there is now several studies, mostly in healthy subjects, going in the same way. So, in conclusion, we know that genetics factors, host genetics factors, and environmental factors are acting on the intestinal inflammation phenotype. There are many data clearly showing that now. We also know that genetic factors and environmental factors have an impact on the gut microbiota composition and function. I did not specifically show you data about that, but it's also very well known that the inflammatory process in the gut by itself will induce changes in the gut microbiota composition. And we also know, thanks to the mouse study, but also to fecal microbiota transplantation study in humans, that the microbiota by itself can have some impact on the intestinal inflammation. And here you can see that there is a kind of vicious circle between the intestinal inflammation and the altered gut microbiota. And this suggests that to be efficient in the treatment of patients with inflammation in the gut, it's true, I think, for patients with IBD. It might be true also for patients with PID with intestinal inflammation. You probably need to target both the immune system and the microbiota. And I think it is reasonable to say that the microbiota in PID might play a role in the phenotype heterogeneity. You know very well that some patients with the same genetic defect will have a different phenotype. So the microbiota, such as many other environmental factors, could be an actor, a player in this heterogeneity. And if it's true, you might think of the microbiota as a therapeutic target to explore. And there are many possibilities to target the microbiota. And of course, fecal microbiota transplantation might be one of them. And this is what we try to do now in conventional IBD, but this might be something to think about in other settings such as PID with IBD-like phenotype. So thank you very much for your attention, and I want to thank all my colleagues from the gastroenterology department in Saint-Antoine Hospital, and all the people in my lab, in both INSERM and INRA, and all my collaborators, and particularly the team of Alain Fischer in Imaging. Thank you very much.
Hi, my name is Kate Sullivan, and I've been assigned to talk about practical approach to diagnosis and management of bowel disease and PID. And you'll notice that the session is entitled Thinking About the Cesspit, and that seems like a terrible thing to be talking about, but I understand that the gastrointestinal tract is just not as comfortable for immunologists. So let's get started. I don't have any disclosures that are relevant for today's talk. And really, this boils down to motility and inflammation. That's what the patients are complaining about. It's really the motility that is their main concern. And I'll just say in passing that the gastrointestinal tract is the largest immune organ in the body. This particular figure is focused on innate lymphoid cells, but unquestionably there's more of our immune system in the gut than anywhere else. So no surprise that immune deficiencies and immune dysregulation conditions manifest with gut disease. Now, I mentioned that cesspit wasn't a very attractive thing to think about, but we as immunologists, we tend to like things very tidy, beautiful flow cytometry plots, and not really talk about stool and intestines. And I'm exactly the same way. I was something of a reluctant convert to gastrointestinal tract diseases, but let's see if we can get through this together. So what goes wrong? It's really all about motility in terms of the patient's symptoms. So too much motility, and that's really what we're going to focus on today. Nausea and vomiting for upper tract motility problems, diarrhea for lower tract motility problems. I'm not going to talk about too little motility, although I'll have a passing discussion of constipation. But gastroparesis is autoimmune almost all of the time. I think there is a signal in our patient population. It's just that it's so poorly understood, I'm not going to talk about it. And the motility is controlled by GI tract macrophages. And this is from a paper that came out a couple of years ago that really transformed thinking about motility. It is a murine study, but I just want to make the point that macrophages can both drive less motility because they can damage the nerves that regulate motility, and they can also drive increased motility. And macrophages are really kind of a mirror of the immunologic milieu, and so they're really impacted by every other cell. So let's get started. So we're talking today about inborn areas of immunity, which I prefer to primary immune deficiencies, but really same idea. And I'm going to be focusing on direct effects, inflammation causing dysfunction and infections. But do remember there are indirect effects. So all the antibiotics that we prescribe do affect the microbiome, which in turn impacts motility. And then just being born with an inborn area of immunity and having a different balance can change motility. We do see a very high rate of reflux, for example, in babies with SCID before they're ever transplanted. And so it's worth just acknowledging that there are other aspects than what I'm going to talk about today. So let's start with infections because it's part of every differential. And this slide could have been 100 pathogens long, of course. I picked the things that, for me, account for about 80% of the infections, but you may have additional infectious landscapes that are important where you live and work. So the diagnostic approach is culture, of course, that's where you start. Remember, PCR is more sensitive than antigen tests for norovirus and rotavirus. You may not have access to PCR, but it is a bit more sensitive. Of course, get an ova and parasite. But the reason I highlighted the 3 pathogens in the previous slide and what I wanted to highlight as a little bit of a pearl is that biopsy can be required. So the first 3 tests are sort of where you start, but recognize that a biopsy can be required for Giardia and Cryptosporidia. Ova and parasite is not terribly sensitive, even when you use antigen detection methods. And do recall that CMV is often tissue specific. So we see CMV colitis without CMV viremia. So you really do need a biopsy, even for infections. I think sometimes people think about the biopsies just for inflammation, but very helpful as a diagnostic approach for infection as well. Now, again, I wish I could talk about more different things, but I picked out Giardia and Cryptosporidia to talk about. I think norovirus is actually maybe more of an issue in many patients, but I think there are a couple of things to talk about for Giardia and Cryptosporidia that I do want to highlight. So Giardia, of course, is more common in antibody deficiency states. Cryptosporidia, more common in T-cell deficiency states, although, again, not a hard and fast rule. And Giardia is not that easy to eradicate. So the first-line agents are metronidazole, ornidazole, tinidazole, and albendazole. First-line agents will get you what you need at least half the time and probably more often. In the general population, the success rate is 75 to 80%. But certainly, I'm here to talk to you about what happens when the easy path forward fails. And so after primary treatment failure, I usually go to dual therapy. Now, in the literature, quinacrine has been demonstrated as monotherapy to be effective for primary treatment failure. So let's say you use metronidazole, you don't get a cure, and so then you can go with quinacrine. But for immune deficient patients, I don't usually do that. I usually go to dual therapy if I have someone who's failed monotherapy. And the reason to know about Giardia is that metronidazole resistance is known. It's not well characterized. This is not plasmid mediated, but there are mutations that seem to be associated with metronidazole resistance. And so as that increases in the general population, it does mean that you will have more primary treatment failures. And the reason I picked it to talk about today is that there are some unusual complications that I think are not especially well known, and at least were certainly a lesson for me, so I'll share them with you. So as is true for any gut infection, you can have reactive arthritis. So reactive arthritis isn't infection with Giardia. It's not that the organism is going to the joints, but do recognize it's a relatively common complication. There are ocular complications, and iridocyclitis is really part of the reactive arthritis spectrum. But in infants with Giardia, there's this very peculiar complication where the retinal pigment epithelium sort of degrades. It's not clear what the mechanism is, whether it's nutritional or something else, but that's seen really dramatically more common in infants than older kids and adults. I've had one patient with hypokalemic myopathy, and I was a little slow to recognize it, so I did want to highlight it here. And then what I really wanted to talk about is post-infectious irritable bowel syndrome. So this is really common after Giardia, and it can be tricky because you'll think that the symptoms are treatment failure, and so very easy to say, well, I'm going to now give you quinacrine and metronidazole or whatever you prefer, rather than recognizing it as post-infectious irritable bowel. And it can last for months. It typically responds to dietary interventions, no caffeine, no spicy food. Some people have particular food sensitivities, but it's usually a dietary intervention, although there are medical management options available. So just recognize that and be sure that you're not over-treating someone thinking that it's just persisting disease. And again, often a biopsy will get you the answer. Cryptosporidia, I want to highlight for roughly the same thing. Again, much more common in T-cell deficiency states. Nitazoxanide is the primary therapy, although it's a bit hard to get, at least in the US right now. And I want to highlight a paper that showed that for CD4 counts over 200, nitazoxanide had a clearance rate of about 70 to 75%, but for CD4 counts less than 200, it was no better than placebo. So there is, at least in HIV, a T-cell threshold, which I think is a lesson for us for our functional T-cell deficient patients. Because of the shortage of nitazoxanide, there's an alternative, which is azithromycin plus paromomycin. But one of the reasons I wanted to highlight cryptosporidia is look at the course of treatment. These are very long treatment courses, and so I do want to highlight that. I think people underestimate the amount of time they need to treat for. There are some alternatives which I've listed there that are at least effective in small case series. So there's data to support them, but much smaller series than for nitazoxanide or the dual therapy that I mentioned. But there's two reasons I wanted to highlight cryptosporidia. One is your patient is telling you that their T-cell compartment is very compromised if they've got chronic cryptosporidia. If you cannot clear them, they're telling you that their T-cells are dysfunctional. And so think about transplant. If they're that susceptible to cryptosporidia, they're going to be susceptible to other opportunistic infections and lymphoma. So do think about whether a transplant is indicated and could actually clear their cryptosporidia. Just as is true for Giardia, there are some late symptoms that are not microbiologic. So arthritis, ocular pain, headache, and then this post-infectious irritable bowel syndrome. And speaking of irritable bowel, we're going to transition now and talk about small bowel disease. And just to orient you to the pathology, so on the left-hand side, I have normal biopsies. On the right-hand side, I have villus blunting. Notice that the small bowel has very long villi and very short crypts. You can see the crypts at the bottom. I don't know if you can see my arrow, but there's just a little bit of crypt down here, and easy to find because of the mucin-producing cells that look clear. Over on the right are villus blunting. Notice the big inflammatory infiltrate there and the very blunted villi. You don't really need to be a pathologist to be able to make that diagnosis. Now, I'm going to lump a lot of things into CVID enteropathy, which is not really fair, but just to sort of broach the topic and get to the point where we talk about treatment, I'm going to lump a lot of things together. Sometimes this is called non-celiac sprue by gastroenterologists, a term that I really hate. But it certainly is often misdiagnosed as celiac disease because celiac disease is 1,000 times more common, but it doesn't respond to gluten removal. Now, I will say there are patients with common variable that truly have celiac disease, so it does happen. But that's not what I'm talking about. I'm talking about villus blunting that's not gluten responsive. And so this is a reasonably common complication, about 1 to 3% of patients, but there's no regionality. As best as I can tell, it's been reported from Europe and Asia and South America. So there doesn't seem to be any regionality to it, which is certainly not the case for celiac disease. Well, when you see this, it's important to rule out infection. In some of the beautiful papers to come from ESID and other European collaborative studies, CVID enteropathy was seen in the patient cohort that was enriched for lymphoproliferation. Now, we know that in this subset, there's clearly patients that have CTLA4 and LRBA deficiency, and that's what's driving the lymphoproliferation and enteropathy. But there's probably also some polygenic cases. So just recognize the clinical constellation. And one of the reasons I'm highlighting this as one of the things to talk about is that it has a huge outsized impact on quality of life. So when you hear a patient say, yes, I'm having 4 to 5 stools a day, that probably sounds like not a big deal. And if they work in an office building with easy access to toilets and they drive to work no more than 15 minutes, it may not be such a big deal. But recognize that for patients that need to take a bus for 30 minutes to go to the grocery store, they probably can't do that, right? They need to be within a relatively small number of minutes from a toilet, and so they can't take a bus and go to the grocery store and be gone for a couple of hours. So you will hear patients bitterly complain about their diarrhea if you can get them to talk about it. Patients, just like us, patients don't really like to talk about their gut and their stools. Well, the mechanism of the villus blunting is T cells. This is the beautiful immunohistochemistry, and it highlights how the T cells are really localized at the surface, and the mechanism of villus blunting we think is T cell mediated, and we think it's driven by cytokine production. So what's the diagnostic approach here? So be really careful to look for pathogens before you invoke an autoimmune mechanism. I'd like to highlight that Helicobacter, which is still very common, is a reasonably common mechanism for villus blunting. If you've had a patient travel to tropical areas for over a month, think about tropical sprue. It is infection, even if we don't know the pathogen. Do get a biopsy of the small bowel. I always check for lactose deficiency, not that it's driving the villus blunting, but because it's a common consequence and it definitely complicates symptoms. If it hasn't been done, I do a gluten trial, and it has to be a real gluten trial, not, oh, yes, I cut gluten out of my diet. And what they really mean is that they stopped eating bread, but they still eat pasta. So you have to really be a good detective and make sure they really understand what gluten restriction is. I'm going to highlight drugs in a moment. And then if you are seeing adults, think about small bowel lymphoma and send T-cell clonality studies. Maybe not at the beginning, but at some point you probably should. So this is from a paper that looked at non-celiac causes of villus blunting. And I'm showing it just to demonstrate the vast array of infections, autoimmune diseases, and other etiologies of villus blunting, because you do have to be a very good detective and not just jump instantly to CVID enteropathy. It's really a diagnosis of exclusion. Well, what do? So certainly optimize nutrition. And I was slow to get on this train. I remember Helen Chappell about 10 years ago started talking about the importance of monitoring nutrition, really being very proactive in monitoring vitamins and micronutrients. And it's absolutely the case for these folks with chronic diarrhea. Important to check both fat-soluble vitamins and other vitamins, because they will lose them when they have fat malabsorption. Ensure that the immunoglobulin dose is adequate, particularly if there's a big eosinophil count in their inflammation. They can have a lot of protein loss in their small bowel, and so you may have to adjust the immunoglobulin dose. But what do we do medically for our patients? So because I think this is a T-cell directed issue, I start with sirolimus or abatacept, because I think it's addressing the T-cell problem at the root. But I certainly have colleagues who like TNF inhibitors and have said that it's particularly good for NFKB1 mediated disease. So I wouldn't say no to that. And we do use it for Crohn's disease with villus blunting. And some people like rituximab. I usually use it for nodular lymphoid hyperplasia, but some people like rituximab. Just be aware that it does have a signal of IBD as a complication. And that brings us to colonic disease. So fairly common in immune deficiencies, again, ranges from about 3% to 50%, thanks to Leonard Hammerstrom for really looking at IgA deficiency as an entity associated with all of the complications we see in other diseases like common variable. So fairly common, in fact, for our very early onset IBD clinic, chronic granulomatous disease is our #1 molecular diagnosis. So it can be the first sign of an immune deficiency. And the symptoms are usually the classic textbook symptoms: diarrhea, blood in the stools, and cramping. But particularly in kids, we see constipation as a presenting manifestation. I wouldn't say you have to biopsy every child with constipation. Just be aware that if the nerves are damaged by the inflammation, you can actually get constipation. Again, let's start with pathology, because IBD is a pathologic diagnosis. So on the left, I've got crypts in a normal biopsy. You can see the crypts are very tall, very narrow, and they go all the way down to the submucosa. On the right-hand slides, I'm showing you IBD. So in the middle, you'll see a little crypt abscess. That's part of acute inflammation. Notice the big neutrophilic infiltrate here. Again, that's the acute inflammation that we look for. And then you must also see chronic inflammation to make the diagnosis of IBD, and that's shortened crypts, branch crypts, crypt atrophy, and decreased mucin production. Now, you can see some other features. So in the middle here is a crypt that's cut in cross-section, and those cells with a clear halo are apoptotic cells, so very common with very aggressive disease in adults, and we see it quite a bit in kids with early onset IBD. So you can see a lot of apoptosis. Also in this biopsy, notice the pink sprinkles in the interstitium. Those are eosinophils. Do not be fooled into thinking that the eosinophils mean this is an atopic condition and you need to think about dietary restriction. Eosinophils are part and parcel of many inflammatory responses. So some diagnostic tips. So infections can look almost the same. So if you open a textbook, it says infections will cause the acute inflammatory response, but not chronic. That is not true for our patient population. I don't exactly know why, but certainly we've seen chronic changes to the crypt with infection. So don't be fooled. So infections, you definitely have to rule out. When you see a lot of granulomas, think about sarcoid. I honestly don't know what sarcoid is. And when I see it in someone's chart, I think, oh, this is going to be a monogenic inborn error of immunity. But there is an entity of sarcoidosis, and so I do respect that. But when I see sarcoid as an explanation for granulomas in the gut, I definitely worry about that diagnosis. For Hermansky-Pudlak, you're looking for pigmented macrophages in the granulomas. That's sort of a good bioassay for Hermansky-Pudlak. When you see ulcers that kind of are very prominent and not associated with the same degree of inflammation, think Behçet's. And then there are several monogenic causes of IBD where ulcers are very prominent. And it's not that there's no inflammation, it's just that the inflammation is less than you would expect for the degree of ulceration. Well, one of the things I want to highlight is drug-induced IBD, because I think it's often quite difficult to kind of get to that diagnosis. Non-steroidal anti-inflammatories are definitely the number one cause. Checkpoint inhibitors are getting a lot of attention, but to be honest, those are going to be picked up by the oncologist, not by us, generally speaking. So I do have a differential for colonic inflammation. There can be anatomical issues. Usually those are not very mysterious. Infections, medications. I've highlighted here mycophenolate because we in our group use a lot of mycophenolate and it does cause IBD. So just be careful about that. And then be aware that biologics actually have a significant signal for IBD. So TNF inhibitors really almost limited to etanercept. Secukinumab, which is used for psoriasis, has a significant signal of IBD. The checkpoint inhibitors I mentioned, and then rituximab, again, has a signal for IBD as a consequence, and we certainly use a lot of rituximab. We, I mean our community. So this is not meant to be read, it's meant to overwhelm you. So the list on the left is the list of drugs that have been at least somewhat associated with IBD. It's just meant to make the point that drugs are a common cause of IBD and you can't really neglect them. There are some pathologic clues that it might be drug-induced, and those are listed on the table in the middle. I have to say, we usually just try to remove the drug and see if we get progress, but there are some clues that you can get from the pathology. So what do we use for treatment? So I've listed these in kind of different tiers. So we, except for the mildest cases, we start with TNF inhibitors. Not for chronic granulomatous disease, but for pretty much everything else. We like ustekinumab, we like IL-1 inhibitors, and I always like to mention nutritional therapy. I think it is undervalued as an approach. So it has a success rate of 80%, which is the same as for steroids, so we really shouldn't neglect it. I think adults find it hard to comply with, so 80% of your calories have to come from enteral nutrition, but kids often feel so crummy that giving them enteral nutrition isn't a problem at all. So do recognize it has quite a good track record with essentially no side effects other than the burden of making it happen. Antibiotics also have their utility. I've had patients get quite significant relief by using triple antibiotics. I list budesonide and salicylate derivatives because we really only use those for the mildest cases or for adjunctive treatment. And then at the bottom, these are drugs that we definitely use, but we're still trying to figure out the niche for them. So tacrolimus, vedolizumab, and notice that vedolizumab takes 3 months to work, so don't give up on it too soon. And then JAK inhibitors we're using more and more, but I think we don't entirely know which patient groups to really emphasize that with. And then you'll notice I did not mention 6-MP and azathioprine. Maybe because I'm a pediatrician, we shy away from those because of cancer risk, but I know they're widely used by adult gastroenterologists, and they would again be on the milder end. They're not heavy lifters, but for mild disease, they can be quite effective. Well, like everyone else probably at this meeting, we're big believers that knowing the gene can help you target therapy, and we do use transplant, we do use targeted therapy. I'll just highlight IL-18 binding protein for NLRC4. I think people are also using it for XIAP1 colitis, so this product is available on a compassionate use or through trials, so don't neglect it. You can also use it to figure out what you really shouldn't be using, or you should at least think more deeply about. So we don't use TNF inhibitors for CGD. I know some people use it as a bridge to transplant. We have transplanted one patient with trichohepatoenteric syndrome with reasonable engraftment, but it's not widely used. And I'll just highlight 2 common scenarios in our population. So XLA, usually it's adult onset. I have seen 2 kids with it. I think it responds well to TNF inhibitors, but my youngest patient actually got a year-long remission from antibiotics. So a year-long remission is nothing to sneeze at. It's really, they were quite happy with it. But my clues to you are be very sure it's not infection and be really sure to monitor immunoglobulin levels. So I like TNF inhibitors for XLA colitis, but I know there are other treatment regimens out there. Chronic granulomatous disease, another common condition where we see colitis. Onset at any age. I mentioned it's our #1 molecular diagnosis in our VEO IBD clinic, but the cumulative incidence goes up with age. It really responds to steroids, and you can really you can get away with steroids in the short term, but you can, at least in my experience, you can never get the patient off. So thanks to a study from the NIH, we use an IL-1 inhibitor to cover the steroid taper, but do recognize that transplant is curative and you don't have to have the IBD in full remission to go to transplant. And then I'll just say there are case reports using vedolizumab and ustekinumab. I haven't used them, but certainly there's literature out there. No gene, no problem. So we all have patients where we can say they have an immune deficiency, but we don't have a genetic diagnosis. So what do you do in that circumstance? So neutrophil abscesses in the colon, sort of the classic IBD pathology, TNF inhibitors, steroids, ustekinumab, IL-1 inhibitors, tacrolimus. If it's small bowel enteropathy, we're much more in the T-cell realm. Again, other people have other approaches, but this is kind of what we like. And then for infants with IBD, we often will start with an IL-1 inhibitor, but we've been using more JAK inhibitors, and we like enteral therapy. It hasn't been as effective in infants as it has in the older kids and adults, but we will often try it. And of course, I'm not a gastroenterologist, so I have to acknowledge the people that have taught me all that I know. Judith Kelson is the director of the Very Early Onset IBD Center. She's taught me much of what I know, and then Maura Conrad has taught me a lot about the nuts and bolts of managing patients. So I'm going to end there and say thank you, and I'm looking forward to the ESID meeting. And if you have questions, you can always email me. All right, take care.
I would like to welcome to this Q&A session on PID and gut. First of all, I would like to thank all the speakers for their great contribution to the session. We have already several questions on the side and from you audience, and Bodo Grimbacher, the other moderator, will gather your question according to the topic and address to the speakers. So the first speaker will be Nadine. So Bodo, to you with the question.
Bodo, you're muted.
Thank you, Nadine, for the wonderful presentation of the classification of the variant forms of PID. Now, many of the questions in the chat actually go towards therapy. And so it's important when you talk about therapy to know what are the cell types making the patient sick. Is it more a neutrophilic infiltrate, or is it a lymphocytic infiltrate, or is it actually the endothelial cells which is sick? For instance, with an endothelial cell disease, hematopoietic stem cell transplantation would not cure. So many of the questions ask, does it make a difference, or how would you treat patients differently, whether there's a neutrophilic infiltrate versus a lymphocytic infiltrate? And how do you do that when working up these diseases on a pathophysiological ground?
Well, it's I think it depends. For the disease with the lymphocytic infiltrates, most of them are more disease affecting, which I have called classified as more autoimmune enteropathy. And for a number of these diseases, we have some targeted therapies like the CTLR4 agonist, like depending on the defect. So I would probably consider the defect which is involved, and as much as we can try to target, in fact, the defect. So for the upper part, some of the defects clearly need hematopoietic stem cell transplantations when it's really the lymphocytes which are sick. When there is no targeted treatment, and when they are extremely severe. For disease which affect more the lower part of the intestine, and where the neutrophilic infiltrate is more important because it's more probably a problem of host microbiota interactions and recruitment of innate cells. It depends. In the case of IL-10 receptor deficiencies, clearly the only treatment is hematopoietic stem cell transplantation, and then one probably has to be careful depending on whether the epithelial cells are involved, as in the case of Nemo, or whether it's only a defect on the hematopoietic compartment. And for example, in the case of XGAP, it seems that you really have to go for hematopoietic stem cell transplantations early on because it's more difficult later on. And then we have heard that for NLRC4 you may block IL-18. So I think it really depends on the disease. Is it what you suggest? Have you other suggestions?
From the other speakers, do you have suggestions, comments?
I thought Nadine summarized it beautifully.
So may I ask? So if you see a neutrophilic infiltrate, does it immediately mean that the neutrophils are the culprit, or are there lymphocytes which are attracting the neutrophil infiltrate?
Yeah. Perhaps I should say that we have not actually considered this way. We have really divided. I think it's simple. We have more divided patients with lymphocytic GVH-like disease and patients who have a disease in the lower part of the intestine with ulcerations and more or less perianal lesions, and not consider directly the questions of the neutrophil infiltrates. So it's more probably neutrophils and also macrophages, monocytes. So I think it's more complex than only neutrophilic infiltrates. But neutrophilic, which means inflammation, while lymphocytic infiltrate would be more a problem with adaptive immunity. So, but it's probably not only the neutrophils. Okay, so it really depends of the underlying defects, I would say. Because we have granuloma in CGD, so I mean, you expect the neutrophils to be sick. So I would be careful to classify about neutrophil infiltrate. I would say neutrophil inflammations, lower part.
Yeah. Other question for Nadine, Bodo?
We discussed. I think we covered many of them. The next set of questions go to Harry, and there was a lot of interest in the audience about the microbiome. One of the questions was, do you know how the biologics, for instance, the anti-TNF treatments, affects the composition of the microbiome?
So there is some data in conventional IBD, so it's not PID. So what is seen is that when you give anti-TNF agent to patients with IBD, you will have your microbiota will change and will tend to go back to something normal. But of course, it does not mean that it's a direct effect of the anti-TNF on the microbiome. The way I see it is that, of course, as I mentioned during my talk, the inflammation by itself also shapes the microbiome. And if you basically cure the inflammation, then the microbiome will tend to go back to something normal.
Can I actually ask a question in this direction regarding treatment and microbiota? Harry, do you see there are any data on what happens during the bone marrow transplantation to the microbiota in these patients?
So there are several data on that, and actually we also work with our colleagues hematologists in Sokol Hospital, not in the context of primary immunodeficiency, but mostly in other settings. The problem is that during bone marrow transplantation, the patient undergoes so many aggressions, antibiotics, chemotherapy, and so on, that giving a precise answer to this question is very difficult. What we mostly see is the effect of the treatment the patient receives, and knowing whether changing the immune system in this process will really impact the microbiome is very difficult, because you have all this massive aggression on the patient between it. Of course, if you look at mice data, you can partially say that there is some effect.
I have one question here, which basically relates also to the statement you just gave and links it to your presentation. Because in your presentation, you said, or you showed data, that the genetics influences the grouping of the microbiota composition. However, may it not also be because different genetic diagnoses get different treatments? For instance, one gets only antifungals and the other ones does not get antifungals, that actually the treatment impairs or is responsible for the grouping this person asks?
Yeah, so in this analysis, actually, we took into account the treatment, although I agree it is still a limited analysis, but we took into account the treatment when we showed the effect compared to the healthy subject.
And then another question towards your presentation was, you showed us the data on card-deficient mice. Now, there are also quite a few of card-deficient humans without really inflammatory bowel disease as a major problem. How do you account for that?
So, card9 knockout mice do not have any spontaneous inflammation in the gut. What we observed is a higher susceptibility to inflammation. So, there have been some data showing inflammation in patients with card9 deficiency, but it was more. Of course, you cannot do the same investigation in humans than the one you do in mice, but it was more related apparently to the fungi overgrowth than to anything else.
Mm-hmm.
Okay, others. I think now we shift to the last speaker, Bodo, or you have other questions?
Yeah, there was one interesting set of questions, basically relating how should that knowledge about the microbiome now be translated into clinics? Because many physicians ask, do I now need to collect the stool of the patient and determine the microbiome, and in what of in which disease does it make sense, and what will be the impact? Would somebody stick out the head and say, if the microbiome is like this and this, I need to prescribe probiotics?
So this is a crucial question, and we have also this question in many other diseases. So my message today is do not analyze the microbiome of your patients now, at least for their clinical interest or your medical decision. Currently, it's not useful. However, of course, I hope and I think that in few years, we will have microbiome-based biomarker to help us to decide maybe the treatment or maybe guide our diagnosis and so on. But currently, I never prescribe any microbiome analysis to any of my patients.
Are there currently research consortia where physicians with interesting groups of patients can submit their stool samples to make them available to the research?
So I guess there is, depending on the disease, there are many consortia. For primary immunodeficiency, maybe Nadine can answer if there is. But for IBD, for example, there are many consortia in many countries. So I would suggest going to your IBD specialist, for example, in your country, and there will be a study for sure.
No, I don't think there is, but I think it would be an excellent idea to try to come together and to try to find for research purpose what we could get from the microbiome studies, because indeed, and to decide how to do it, because there is clearly a question of egg and hen, so chicken and egg, and so, but there are now so many evidence that the microbiota can influence the immune system in and out of the gut, that I think that we should probably try to come together and to find a way to understand.
If I may, there was one question I saw which I think is important regarding probiotics. Someone asked whether I can give probiotics to a patient with primary immunodeficiency. So I think that if the probiotics goes into the gut, it's not a problem because, as I mentioned, there's trillions, billions of microorganisms in the gut. So adding few more, it's not a problem. The problem that has been linked to probiotics is if you have extraintestinal diffusion of the microorganism, notably with a catheter, for example.
Can I forward these questions to Kate? Kate, is it always safe to give probiotics to patients with, for instance, impaired immunity against fungi? And the probiotic may contain fungal components, live fungal components?
Well, we're talking about 430 different inborn errors of immunity, and probably 100 of them have susceptibility to some kind of enteropathy. So it's hard to generalize. I would say I wouldn't give it to someone with SCID. There actually are some limited data out there, and generally speaking, in immune-compromised patients across a whole broad range of categories, probiotics are safe, and I think many immunologists do use them. I would say one caution is it ramps up the likelihood of adverse events if you're giving them by G-tube or some non-standard way. So remember, they're encapsulated for oral ingestion, and when you start giving them in other ways, I think you do introduce an increased risk of adverse events.
Interesting.
Kate, does the composition of the microbiota so we're talking about microbiota, and the probiotics are very, very variable. So some are killed, some are alive, some contain fungal components, others don't. So can you please specify a little bit more which are the safe ones and which one you would stay away from when a patient is a combined immunodeficiency patient or a patient who suffers from CGD?
Well, I'm going to just cut to the chase here and say no. There are no data that are doing any kind of comparative analyses of different types of probiotics. And I don't honestly see the sense of using killed bacteria, although I do sometimes prescribe bacterial extracts, which I think are very popular in Russia and South America, but not in this setting. I use them in a different setting. So I'm just going to cut to the chase. There's actually no data. Would I be nervous about using probiotics with fungi and CGD? Yes, but that's not usually a setting where I would be thinking about probiotics.
Okay, Bodo, do you have other questions?
There's another question for Kate, and this participant wants to know more about the role of vedolizumab in a subset of patients. Do you use it a lot, Kate?
I would say about 10% of our cohort is on vedolizumab. So I'm speaking now about the entire early onset IBD cohort, not just the monogenic cases. So we do use it. We do like it. I tend not to think of it as a very heavy lifter. So it's not going to usually get someone out of crisis, for example, partly because it has a very slow onset of action. So about 3 months. And partly because I think, although it is changing the recirculation of T cells, I don't think it just has as much heft. So if I've got someone where where I want to go down a lymphocyte-directed pathway, and Nadine talked really beautifully about how you would sort of choose more of a lymphocytic approach versus a neutrophil inflammation approach. So if I'm trying to go down the T cell approach and someone is very, very sick, I will either add vedolizumab plus a JAK inhibitor or vedolizumab plus something like tacrolimus if I really need to get instant action. So I just don't think of it as a heavy lifter, although I do think there's an important role for it, and it can be very effective.
You mentioned the JAK inhibitors. What makes you think in IBD patients to use the JAK inhibitors?
Well, Bodo, you have some data about JAK inhibitors. So we like them a lot. To be honest, we started using them in infants because we're so limited in terms of what we think we can safely use in infants, and our pharmacy really restricts us. So we started using JAK inhibitors in infants before there was really any data on the global IBD population. But boy, are they effective. They're quite impressive. And so we have started using them more and more over time. And now we will use them for small bowel enteropathy, which was not a direction we started until recently.
And do you do an interference signature before using them?
We do not. We have access to it. It is run in our hospital, but we don't. I don't know whether it would be predictive of effect or not. I'm just not sure.
And can you comment on vedolizumab versus abatacept? Because, abatacept generally blocks T cell activation also in the gut. And is there a difference between using abatacept, which probably should also diminish the T cell activation in the gut, as opposed to vedolizumab?
That's really a great question. I have mostly used abatacept in patients with known CTLA-4 or LRBA. We have only one patient where, out of desperation, we tried it without a known gene defect. So I think that's a great question. You're right. It does make some mechanistic sense to think that if you want to go down a T cell path, abatacept might be a nice direction to go, and certainly a kinder strategy than tacrolimus. So I don't personally have an experience outside of CTLA-4 and LRBA.
So how did that one patient turn out?
I'm sorry, I didn't hear you, Bodo.
How did that one person without CTLA-4 turn out on abatacept? Did it help, actually?
Yeah, actually did great.
Mm-hmm. That's interesting.
Can I ask a question? It's a little bit outside of the treatment, but I heard Nadine mentioning a very hot topic nowadays, the multigenetic inheritance of this disease. So given that somehow they're very complex, and despite you try to give a very nice clusterization according to the genetic involvement, the type of gastrointestinal tract and whatever, sometimes there are lots of overlapping features. So this question is more for Nadine and Kate. What do you think in the future we should actually maybe try to look into multigenetic pathways, or do you think this is something that really doesn't have that important role?
Who wants to? Well, personally, I believe that we have a lot to learn from the monogenic disease, perhaps to help stratify patients with more complex disease. And it's clearly something that we would like to do, is to try to characterize using new technologies type single cells and so on, to try to establish the pathway which are modified by specific monogenic disorders, and then to try to go to more complex disease, to try to help this complex disease where finding the right treatment is at the moment very difficult to decide which treatment you should use first. So that's how I see the thing.
Nadine, one more question regarding research. Does research with organoids help in this field?
Organoids, we use it more in the lab for disease which affect the epithelium, to try to understand the mechanism of how to decipher the mechanism involved in the differentiation. Organoids, apparently, and we use more IPS-derived organoids, in fact, for reasons which are linked to the fact that our children are difficult to biopsy. But people use indeed intestinal organoids, and it seems that in IBD they keep the epigenetic marks, so it can be useful to try to understand and to decipher the role of the epithelial cells and perhaps the epigenetics. I must say that, and also to try to understand the interactions between organoids and lymphoid cells. More and more people do that. The problem with the organoids derived from the intestine is that you have only the epithelial layer, so you will miss the interaction with the stroma. If you use IPS-derived organoids, you don't have all the epigenetic marks. You have the stroma, but you have a problem is that you cannot get the same type of differentiations that the regular the organoids that you get from the intestines. So at the moment, I think it's an interesting issue, but to be
Eleonora, thank you, Nadine. Eleonora, do we have time to pop the norovirus question?
I think we are really running out of time. I don't know if the question is quick and the answer is super quick. I think we can.
So then we go for Kate. So what is the current approach for norovirus management in our patients? Quick answer.
I saw that question come in. I was hoping we were running out of time. So this is what I'm going to say, and I don't know if it's right. Everyone knows there's no controlled studies, and I'd be interested to hear what others do. I actually come at it from the side of inflammation. So along with Harry's idea that there's this interplay between the immune system, the pathogens, the commensals. So I actually attack the inflammation, and weirdly, I tend to treat it with sirolimus. And I can't say I'm 100% successful, but I certainly have achieved some clinical benefit that way. And so that's what I tend to do.
Perfect. Okay, I think it was a very interesting and lively question. Thanks to all the speakers, to all the audience for the very enlightening question that fostered the discussion. Thanks to you, Bodo. And I think this is it. Bye to everyone.